Hello, good evening. Please take a seat. Welcome to the Italian Culture Institute. And thank you for joining us for this very exciting, yes, I would suggest you take a seat though. We have plenty of seats here also in the front if you like. Celebrating the very first edition of Italian Research Day in the world, an initiative created by the Ministry of Education, University and Research to promote the important work of Italian scientists around the world. Before we start, I'd like to say that the Italian Consul General Antonio Verde cannot be here tonight because of some prior commitment, and so is the Consul General of France, but both of them, they're sending to Emiliana their best regards, and to all of us, of course. And we have a representative from the Consulate General, Patrizia Payar. The Italian Culture Institute of Los Angeles has invited Emiliana Borrelli, Professor of Microbiology and Molecular Genetics at the University of California, Irvine School of Medicine, to discuss a research activity on the neurotransmitter dopamine and its role in brain diseases, movement disorders, and addiction. Her work has earned her international recognition and prestigious awards, including the title of Knight of the National Order of the Légion d'Honneur, the most prestigious French civilian award, and the Golgi Medal Award in Italy in neurosciences from the Golgi Foundation. It is an honor to have you here representing the very best of Italian science and present you with the Italian Culture Institute Los Angeles Creativity Award. Thank you to all of you for attending tonight and participating in this very first edition of the Italian Research Day. This evening event will begin with an introduction by Michael Stamos, Dean of the UC Irvine School of Medicine. Under Stamos' leadership, the School of Medicine recently received the highest accreditation possible from the Association of American Medical Colleges, AMA, Liaison Committee on Medical Education. He has led the school's faculty cluster hiring initiative and has been a key partner in developing and executing the UC Irvine Health Strategic Plan. As a courtesy to others, I'd like to ask you kindly to turn or silence your cell phones now. Thank you. I would like to invite you all also next week on April the 24th at the Italian Culture Institute of Los Angeles, for those of you who love Italian cinema. Will be the very first edition of another Italian, I know we have so many here in LA, Italian Film Festival, which is called Cinefest Italia in Los Angeles, but actually is the film festival in Santa Fe that will be opening this very weekend, and we'll have the Italian directors and actors that will receive the awards there. So we have the winners here at the Institute next week. Now please join me in welcoming to the stage with a very warm round of applause, Dr. Michael Stamos.
Thank you very much. It's really a distinct pleasure to be here tonight to recognize one of our real stars, Professor Borrelli. I'd like to say just a few words about UC Irvine, because UC Irvine is not as well recognized, particularly in West LA, as other UCs. I actually started my career in the UCLA system, spent 11 years and then moved to UC Irvine 16 years ago. And I'll just say that I was drawn to UCI at that time by its audacious vision for really a team approach, patient-centered approach to care, which now has become very, if you would, expected and standard, but back 16 years ago was not. And I think that it's been a very wonderful journey for me. I never aspired to become the dean, but when I was asked to step in about 2 years ago, that position, I did so willingly, and now with great passion and energy and enthusiasm. And it's really because of the great stars that we have on our faculty and the opportunity to really shine a light on all of them to show their successes. The world looked very different 16 years ago when I got to UC Irvine, and technology has impacted all of us in so many ways that are so unpredictable and different than it was 16 years ago. But we've embraced it through all of our missions at our campus, including our iPad curriculum, electronic health efforts, et cetera. But also education, research, and healthcare in the 21st century is all about teamwork. And really, I'm pleased and honored to be able to implement this team approach as part of our strategic plan. And a big part of our teamwork is our basic scientists. And you heard mention of our cluster hire program, and that really has to do with matching up and pairing our great scientists with our great clinicians, to build a team around those scientists to help translate that great work that they're doing into the clinical infrastructure of our environment, to make sure that patient care is impacted in a positive way by all that great work. And I think the work that you'll hear about tonight from Professor Borrelli fits well into that. We have many faculty stars. We have 800 full-time faculty, and we have many of our faculty have received many awards and many accolades. But I'm not going to spend any time on that. I'll just say that we have recently made, and we will make additional strategic investments in people and infrastructure. And most specifically, we have recently made additional investments and provided resources to Professor Borrelli and her team. And we expect, therefore, what you'll hear tonight is amazing, and we expect even more out of her and her team. And I know that she won't disappoint us. So you heard a few words already about Professor Borrelli, and I'll just add a few things to really highlight what she's done. She's a Chancellor's Professor at the University of California, Irvine. She's also Director of INSERM U904, which is the only research unit that INSERM has established outside of France. The brain controls all aspects of our everyday life. We know that. It directs our responses to environmental stimuli, and this control is exerted through really intricate interactions between molecules, of which the most studied are neurotransmitters and neuromodulators. Dopamine was the major brain catecholamine, is responsible for the regulation of a wide variety of critical functions, including motricity, pleasure, learning, and reward. And that's really been a focus of Professor Borrelli's career. Her achievements have allowed the elucidation of a remarkable variety of molecular mechanisms relevant to the fields of neurology, psychiatry, and neuroendocrinology. In this respect, her work on the regulation of signal transduction by dopamine represents a notable paradigm. Her most recent discoveries include mechanisms governing dopamine-mediated brain circuits relevant to neuropsychiatric disorders and addiction. Obviously, an area of intense interest right now, given the opiate crisis that we're experiencing in this country, including in Orange County and LA County as well. She's a prominent molecular and cellular microbiologist neurobiologist who has pioneered the use of genetically engineered animals to reveal mechanisms of behavior, drugs of abuse, neuropsychiatric disorders. I could go on and on, but in order to finish tonight and still have time for our reception afterwards, I'll just say that she's been recognized with several awards, and you heard about a couple of them, but I'll mention a couple of others. A member of the European Molecular Biology Organization as long ago as 20 years ago, the INSERM Prize in Physiopathology in 2002. You heard already about the Knight of the Legion of Honor from France last year, the Golgi Medal you heard about, and then also the Umesino Lecture Award from the Salk Institute in 2014. So without any further ado, I would wrap up my part of the presentation and get to the honoree. Not yet.
We'll say a few words before about this prize, because it's a brand new prize, but it used to be here in the past, and it was given before, before Emiliana. Only two other people got it, and one was the late Dulbecco, Nobel Prize for Medicine, and the other one was Andrew Viterbi. So the last one was, I think, 2010 or something. So on this very special day and occasion, Italian Research Day abroad, we decided to give this very award to Emiliana for outstanding achievements. And the award is custom-made for the Italian Culture Institute by a wonderful artist, and I don't know, maybe some of you had a chance to meet him last year. Emilio Cavallini is an Italian artist and designer, and it's custom-made for the Institute and for also this special occasion, and it is inspired by the Pantheon ceiling in Rome. So now, without any further ado, it is my greatest honor to give this award to Emiliana Borrelli.
Okay. So, thank you so much, Valeria, for the introduction, and also to Michael Stamos. I am very honored to be here today and to be the recipient of the Italian award for the Italian Research Day in the world. I'm really honored of that. So, I think you should have waited to give the award after my presentation, but now it's done. Okay, so before I wanted just to give you a few slides that tell you about my life. So I was born in Naples, those are my parents, and as you can see, this is a picture of the beautiful bay in Naples. This is the Vesuvio in the back. This is my sister Annalisa. And for me, it's really important today because today would have been my mother's birthday, and I think she would have been really proud of me. Thank you so much also for that, although it was not done on purpose. So I graduated in biological science from the Università degli Studi di Napoli, Federico II, that you can see is a structure that was built in 1224, and was the first publicly funded university. This is the front part of the university, but most of the time I spent it in this building in Via Mezzocannone, where I was a student. While I was a student, I met this man that is also here, and that, believe it or not, he had here. And we both shared our passion for biology. And we were pretty good students, but we were, most importantly, very well advised by our professors. And they told us that in order to become good scientists, we had to have experience outside in the best institute of science. And so we moved to Strasbourg, where you can see here, this is the cathedral, which is a beautiful Gothic cathedral that is there. But there is a second cathedral in Strasbourg, that is this one, and is the IGBMC, and is the Cathedral of Molecular Biology. And this man, Pierre Chambon, was at the time the director. He is one of the most important figures in the field of molecular biology, and thanks to his studies, we can now understand so many things on gene expression and transcription. I spent 3 years as a postdoc in his lab from 82 to 85, and then I went to Ron Evans at the Salk Institute, which is another amazing structure. Valeria was mentioning Renato Dulbecco. He was there at the time, together with Francis Crick and other Nobel Prizes, and for us was a great environment to develop and to further improve our knowledge and interest for science. So in Ron's lab, I spent like from 86 to 88, so 3 years there. These two guys at the time I moved in Ron's lab were not really related, but they became related, not thanks to me. And they were awarded in 2004 both the Lasker Award, which is one of the awards that precedes the Nobel Prize, for their studies on steroid receptors. So then my career, so while I was in Ron Evans' lab, I applied for a position at the INSERM, which is the National Institute of Health and Medical Research in France. And this institution trusted my capacity and gave me a position for life that I accepted, although I started in 89, so I got my position in 87, but I started in 89 my own lab in Strasbourg at the IGBMC, where I started as a research scientist, which is the equivalent of assistant professor. In 94, I became associate professor or research director class 2. And then in 2000, research director class 1, which was the equivalent of full professor. And then I moved to Irvine in 2006 as a professor in the Department of Microbiology and Molecular Genetics. And I have to say, I put this slide also because I need to thank the INSERM, because the INSERM has been funding me all through my career, and also right now, even in the United States, like Michael Stamos said, I am a director of an INSERM unit. This is the only INSERM unit outside France. So my whole career has been based on the study of dopamine. This doesn't tell you much, but this is the structure of this molecule. It's a very abundant molecule in the central nervous system, and it's very well studied because dopamine controls many important functions in our life. In this slide, I just wanted to show where the dopaminergic system in our heads and is at the basis of the brain. So, on this slide, this is one of the few slides that I use in my seminars. You can see here that in red here are shown the dopaminergic pathways. And you can see that, and this is a brain of a rodent. So the dopaminergic neurons are located in the middle of the brain, the mesencephalon, and then from there, this is a very few neurons, but then these very few neurons send the projections everywhere in the brain, and you can see here from this red line. And in doing so, dopamine controls very important functions that range from the control of movement to motivated behaviors such as eating or drinking, but also pleasure, learning and memory, and also reproduction. So, in fact, dopamine is very much associated with our survival because it's important for movement, feeling pleasure, and reproduction. So, because of this involvement of dopamine in pleasure, and so in the media, and here we are in Los Angeles, so this is a movie that has been made, which I do not advise anybody to see because it's quite boring. But it's I forced Paolo also to see this movie because of the trailer here. It says a San Franciscan computer programmer falls in love with a school teacher in the days after the dot-com boom. One of the movie's very good qualities is that it takes time to ponder this issue, whether love is only a chemical reaction or if it is something more. And we are not going to solve this tonight, and the movie for sure doesn't do that, but just to show that, so because it's involved in pleasure and love, so that's why it attracts so much attention. And in preparing this PowerPoint presentation, I also found this slide on the web, which I don't think has any significance scientifically, but it says that extrovert and introvert differ for their sensitivity to dopamine. And in fact, someone that is extrovert doesn't need needs a lot of dopamine, while people that is introvert, it doesn't need much. But as I said, those are just things that you can find on the web. Now, going back to serious stuff, this slide shows that because dopamine is involved in so many important functions, it is clear that dysfunctional dopamine system is involved in many human disorders. And in particular, in Parkinson disease or schizophrenia or the attention deficit hyperactive disorder. But dopamine is also involved in addiction to drugs of abuse, as well as in depression, obesity, epilepsy, and so many other. So, how does it work? This molecule, so dopamine is produced by a certain number of neurons, but in order to work, this molecule has to be recognized by proteins that are present on the membrane of the cell. And these are called dopamine receptors. You see that there are 5 different receptors that exist, but the most important are the D1 and the D2 receptor. And in particular, my lab has been involved in the study of D2 receptor. These 2 receptors are so important that if you generate animals that don't have either one of them, either the D1 or the D2, so double mutants, they don't grow, don't eat, cannot move well, and they die after the second week after birth. So, which means that this really tells you how important is dopamine for our survival. So now, what I wanted to do is just to give you just a few slides to give you an idea of how important the level of dopamine in the brain are important to regulate important function. So the first thing is, for sure, Parkinson disease. Parkinson disease is due to low dopamine in the brain, and the reason for that is because there is a degeneration of the dopaminergic neurons. So not much dopamine, and what happened here you can see on this scan using some ligand that are radioactive. You can see that the healthy volunteer, this red dot that you see here corresponds to how much dopamine is in their brain. You can see that they have a lot of dopamine because red is the highest level, and you can see that in Parkinson, dopamine is almost gone. Parkinson Facts. So in the US only, there is 1.5 million people affected by the disorder, and there are 23,000 people that die from the disease each year. And at the same time, each year 50,000 people are diagnosed for Parkinson, which is it was higher in men than in women. And 1 in 10 people are diagnosed before age 50. So you can see from there that this is a very important disorder and that we need to do anything to understand how this works. And so the phenotype of people with Parkinson includes tremors, and in particular also resting tremors, and then a gait that is very particular. People is unable to move well, and they do small movement. So what I did in my lab was to generate a model by studying the dopamine D2 receptor in a mouse. So although you may laugh because we cannot compare a mouse to a human, but I'm telling you the brain, the mechanism that govern a mouse brain are very similar to that of a human brain. And so using mouse models, what we did was to generate animals that don't express D2 anymore in the brain. And what we found, with our surprise, is that these mice, you see here is the distance traveled. This is a normal mouse. This is the mutant. You can see that these mice don't move well, and they cannot coordinate their movement. So they do have a Parkinsonian phenotype. Having a mouse model for Parkinson allows you to do studies, and this is one of the most important things that I have done in the lab, to generate this model that allow to understand how and what are the mechanisms that govern dopaminergic neurons and dopaminergic function. And in particular, Parkinson disease is treated in humans with giving people a precursor of dopamine, which is the L-DOPA. L-DOPA works great, but after a period that is called the honeymoon, that lasts for 5 to 10 years, having every day this high amount of L-DOPA generates the fact that many receptors are stimulated, and then people start to develop, just a minute, dyskinesia. So dyskinesia are movements that are not very well controlled. And so, again, with one model in the lab, and always this model of Parkinson disease, we were able to show that blocking an acetylcholine receptor in the brain, in particular the M1 receptor, we were able to prevent dyskinesia. So this is a very important finding, and in fact was published in one of the best journals in neurobiology, which is Neuron, because it is important that by understanding how the system works and how dopamine activates particular brain areas and how pharmacological treatment together that can be given together with L-DOPA may prevent the side effects of, for instance, in the case of Parkinson, of L-DOPA. Now, the other important part in which dopamine is involved is addiction. And this is due to too much dopamine. So why addiction? We can get addicted, and you know that, almost to anything. We can get addicted to internet, to the iPhone, to shopping, to work, to love, to sex, to whatever. So why? Because all this activity generate an increase of dopamine in the brain. And when dopamine goes high in your brain, you feel rewarded, you are so happy, and so it's so pleasant that you want to get more. And then, of course, you get addicted. And in fact, those are the 3 steps of addiction. The first one is where is the voluntary take. Then you have a second step where 15% of the population can become addict once exposed to drugs. And so, on the second step, you lose control over the intake, and the habit gets established. And then the final step is when you take drugs without even willing to do that, and you are totally addicted. Can we study this in the mouse? Yes, we can. And in particular here, I just want to show you a cartoon just to give you an idea of what we do in the lab. So this mouse, for example, there is in this test is a preconditioning phase in which a mouse is put in a compartment that is divided in 2 areas, one with stripes and one with checks. And so the mouse is put there, and it will explore the 2 areas without any preference. But then what we do, we start to give the mouse a conditioning. And so we start to give the drug, for instance, in the striped compartment. And so the mouse experiments in this area the feeling of pleasure, because he's getting the drug, he starts to move, he feels happy, and so he will associate the stripes to the drug and to the pleasure. On an alternate day, we give a vehicle, and we enclose the mouse in the room with checks on the floor. So the mouse will very easily understand that the checks, no pleasure, who cares? Okay? And then on the last day, we do the test. And so when we do the test, we remove the separation between the 2 areas, and the mouse at this point is free to choose. So we put them here, for instance, but you can bet they all go to the compartment where there are stripes on the floor. So in this way, it is possible to study the rewarding properties of drugs. And we have applied this to study how, for example, the mice that we generate in the lab that don't have one of the dopamine receptor, the D2 receptor, how they respond to cocaine, to morphine, so to opioids, to cocaine, and to any other kind of drug of abuse. And on this one, this is a video track. So mice are tracked with a video system, and you can see, what I just told you, that during the preconditioning, mice spend the same amount of time in the 2 compartments. And this is the trace that is left by a mouse that has been conditioned with morphine. You can see that they spend way more time in the drug-associated compartment. However, drug of abuse are not the only substance to which we get addicted. We can get addicted also to natural rewards like food. And in fact, obesity is now considered a sort of addiction to food. And again, why? Because whenever you eat salt, sugar, or fat, there is the happy chemical that is written here. This is dopamine. gets high in your brain, you feel happy, you eat more, and then you cannot stop eating, and that's how you can develop obesity. And next, I will show you a very small movie that we have made in the lab with one of the mice that we have generated. And you have to know that mice love peanut butter. It's very fat, but it's very good. So in this movie, you will see the behavior. No, you won't see. Okay. I have to go like this. Yeah. So this is a normal mouse, and in this little tray here, there is the peanut butter. So you can see that the mouse explores, looks at the peanut butter, tries, likes it, and starts to eat the peanut butter. And it will spend, sometimes, and then eventually it'll go. And then next you will see a mouse that doesn't have D2 in one area of the brain that is important for, this is the mouse. And you can see that this guy is crazy about peanut butter. And we can measure how much peanut butter they eat, and they eat way more of the normal mouse. You can see that he will not stop leaking the little tray. So, which well indicates that this receptor that we study is really important in defining the rewarding properties, in this case, of food. And so, again, these mice are a great tool to continue our studies and to understand and to delineate the mechanisms that lead to this kind of behaviors. And then, as a final thing, I want to show you the last disorder that is also associated with dopamine imbalance in the brain, and this is schizophrenia. Schizophrenia, again, facts about schizophrenia: 1% of the population is subject to schizophrenia, which means 70 million people worldwide, a huge number of people. What are the causes of schizophrenia? Not very much known. Schizophrenia is high level of dopamine in the brain. It's treated with antipsychotics, and the antipsychotics are mostly directed against the receptor that we study, so they block this receptor. But nothing is known at present where these receptors are blocked, what are the areas that need to be blocked in order to get the beneficial effect of antipsychotics. And so the most recent theory of schizophrenia is not like a whole level in the brain everywhere that is high, but actually more recently it has been determined that schizophrenia is due to an increase of dopamine in this part of the brain called the striatum, and a decrease of dopamine signaling in the cortex, in particular in the prefrontal cortex. And we do have a mouse that simulate, again, this type of imbalance. And so what we did with this mouse was to analyze gene expression. And this is, I think, my last slide, or one of the last slides. So here, what you can see is just by looking at gene expression in these animals, you see this is the mutant that has a schizophrenic phenotype, and this is a normal mouse. And you can see the color indicates the expression, the level of expression of these genes. And you can see that in these mice that are mutants, all the genes are blue, which means they are down-regulated. And we found 2,000 genes that are down-regulated in this model of schizophrenia, indicating that the level of dopamine is really important in determining the program of this area of the brain. So, at the end, just to conclude, what I wanted to say is that it is important to make this study. Now we have found 2,000 genes, but for sure we cannot pursue the study of 2,000 genes, and we have to find what are the genes that are really important, because you can either block or stimulate them, and it would be good to have only a certain and the most important one. Otherwise, we should make like huge pills that will be pretty scary to take. So the future aims of my lab are aimed at understanding the mechanism underlying dopamine-linked diseases, which will lead to the development of novel therapeutic strategies. And so at the end, I want to thank you for your attention, and I want to thank Dr. Valeria Rumori from the Institute, the Italian Institute of Culture for this award, to Dr. Michael Stamos and Larry Wenzel to come here and be here for me in this occasion. The INSERM, as I said, my family, Paolo, Serena, and my friends, Paola and Laura, and my collaborators that are also present here. If you want to talk with them later on, there is Robert, Stefano, and Marcello. Thank you for working and to improve our knowledge. And then finally to the mice that have dedicated their life to make us understand what is going on. Thank you.
Thank you once again. She's been wonderful. I mean, so simple for all of us that we know nothing about that, but we learned a lot. And she is willing to take a few questions from our audience.
Thank you, first of all. Anyway, thank you, first of all, for coming to us and presenting us with great information. I was just wondering, with the D2, have you studied the genetic test of it? If it lacks in someone's mother, does it lack in the child, or how does it go on through generations, or is that part of
not in my lab, but this is something that is pursued because, for instance, also the epigenetic, it's very important. And in fact, for example, in this last slide, I don't know if you can hear me, in the last slide that I show you where there is this huge difference between the mutants and the normal mice, this is due to epigenetic variation. So when you have high dopamine levels or low dopamine levels in some areas of the brain, you can change their expression simply because you have this imbalance. So the genes on their own, they're okay, but then there are these variations. So it is expected that in the progeny, for example, and something is known, for instance, the kids of women that are addicted to drugs, they have problems when they start learning, when they go to school. It's much harder. So for sure there are epigenetic or messages that go from one generation to the other.
My question is, I have Parkinson's, and I have been told that there are certain foods I should not eat because they are high in dopamine. If Parkinson's is caused by a lack of dopamine, why shouldn't we be replenishing it by the food that we eat?
I'm not sure I understood.
She said that she has Parkinson's, and she's been told that she should avoid certain foods that have high dopamine levels or produce high dopamine levels. Why is that if there's a shortage of dopamine?
Maybe because you don't want to go above a certain level. Because the problem, for example, in the treatment of Parkinson, when you give L-DOPA, you are making more dopamine. But this dopamine will activate any type of receptor. And so this is a problem. Because, for instance, I showed you that our mice that don't have one of the receptors have a Parkinsonian phenotype. So it would be much better, instead of increased dopamine, to have only one, something that is directed only to activate these receptors. So maybe this is the reason they don't want too much. Too much is not good for you. Maybe. I don't know.
Hello, Dr. Borrelli. Thank you for your presentation. A quick question. What other type of medication have we found as a result of this type of research? You know, you mentioned Parkinson's, but you mentioned a host of other addictions and things like that.
But you know that those are preclinical studies. So what we do in the lab is just to show in animal models that it can work in the right way. But then in order to go into the treatment, you know, like there are many years to go by because each one of these drugs have to be tested in humans. And so the way is very long. Sometimes, for instance, we have participated in one compound for Parkinson. We published the study in 1997, and it's been accepted in Japan last year. So it took 20 years. So what we do is important because once you know the mechanism, you can kind of generate something that is more selective, that would activate only one particular type of protein, but then from what we do in the lab to what is translated in medicine, then it takes a long time, which is good because we have, you know, you have to make sure that in humans will work exactly well and there are no toxicity and things like that we cannot estimate in a mouse model.
Knowledge about the impact of other functions in the body that might degenerate dopamine, like if there's a lack of sleep and if dopamine levels are impacted, or do they rebuild during sleep? And
sleep has been also dopamine is everywhere, and so it has been associated with many different things. So it's hard to say no, it doesn't have anything to do, because also it could be an indirect effect that dopamine stimulates an area that then can stimulate the one of sleep. But for example, dopamine, the Parkinson is also characterized by problem of the gastrointestinal tract, and in fact, for example, also for pregnant women, the vomit that when they are pregnant are treated with domperidone, which is a D2 antagonist. So the importance of dopamine in the gastrointestinal tract is also very important. And so, in Parkinson, people experience also side effects, many things.
Yeah.
Hi, thank you for coming. So my girlfriend is actually a neuroscience major in college right now, and we're both very interested in social media's effects on the human brain, and I was wondering if you'd like to share your thoughts on that. I know that they're just social media companies, these apps, they're trying to just get you to crave the next hit of dopamine. And I know you study some about addiction, and I was wondering if you'd like to share your thoughts on that.
For social behavior, you mean?
On social media. Yeah,
social media. What social media? You can find anything in social media, you know that. And so you have to be very selective. For example, this slide that I found on extrovert and introvert and the level of dopamine in the brain, or the sensitivity of these people to dopamine, I don't know how they could have measured that, just based on the knowledge that we have. I am not sure.
I'm referring to some of these companies they are trying to get you to get dopamine rushes from getting likes, and
dopamine, you don't need dopamine. You need your own dopamine. You don't need any external dopamine, that's for sure. There are things that you can do, like like when you exercise or in your normal life, or like natural reward, like I don't know, something that you eat that you like that will generate dopamine. So I would not take myself anything that would add dopamine to my brain. This for sure.
All right, thank you.
Hi, thank you for telling us about your research. I just had a question. I know that you came from Italy to work in America, and my professor was telling me that there's an issue in Italy where a lot of people get like a really good education and they'll leave the country and find work in other places. And I was wondering if you know about any opportunities for people who are looking to go to Italy to work, or like what that would look like in terms of the science field.
Well, science, I think there is no limitation in any way. You know, like you can go where, the only thing you have to make sure when you want to have a good training is to go in a good lab. And the way to know what is a good lab or a good university is based on the professors that work there and on their CV. You can look at their CV. Those are things that you can decide right there whether they're good or not. And of course, there are countries that have more funding, for example, for research than others. But in most Occidental countries, I think you can do good science everywhere. And the only thing that you have to do is to pick up the good places. And the good places is not the place, it's the people that work there that makes a place good. Yeah.
Just a question, because we are all getting old. Bad, but happens. Do the level of dopamine change with age?
They do a little bit, but there is nothing we can do, unfortunately. You know, like you can take drugs, but I'm not sure that you get much better.
Thank you, Dr. Borrelli, for coming here. Excellent presentation. I'm an immunologist, and I was trained here at UCLA in the good lab. John Fahey, perhaps you know him. No, just to say that, in response to what you were saying. My question is relating to the work by Claudio Franceschi from the University of Modena, an immunologist also, whose work demonstrated that the lymphocyte, the immune cell, is what he called a circulating brain. So my question to you is, what is the relationship between dopamine in the brain and the immune system?
Well, that's a very important question, and I am not an expert in that area, but I know that, for example, dopamine can, I think, has an effect on lymphocytes, but I think that everything is connected, and the more we go on with our knowledge of the different systems, the more we know that metabolism and the brain, how does it work, and the bacteria that we have in our guts, they can change the way our brain works. So for sure, the immunology, it's an extremely important part of it. But this would require, and I think there are, I'm starting to see papers where people is doing the association between particular, phenotypes with the brain and the immune system, and I'm sure that they will find a lot of things there. Yep. Questions from friends are not allowed.
Difficulty are allowed. Now, you said before that, for example, for Parkinson, men are more subject to Parkinson than women. Mm-hmm. To have Parkinson in the future than women are. So my question is, does it mean that, generally speaking, men have a lower level of dopamine than women?
But
Men have a lower...
But this is, again, is another area that, those are statistics, those are epidemiological studies based on simply the evidence that it's more common in men than in women. Now, what is the contribution of hormones in all that is not yet completely elucidated. We don't know yet. But clearly there are disorders that are more common, and I think also schizophrenia is more common in men than in women. Yeah, they're weird. We know that. Yeah. Thank you to you.